Okay, I will start. I'm Hector Toala, and I am a meta student in the UCSG, and I talk about SLC25A32 mutations and Riboflavin responsive exercise intolerance. That is that was no definition. Multiple acetylcoenzyme dehydrogenation deficiency is an inborn error on metabolism with a frequent muscle involvement. A short video. That is the sequence of the chromosome, the genes of the disease of the carrier 25, mitochondrial folate carry. Well, that's it. Causes. This deficiency is due to defects in the electron transfer flavoprotein genes ETFA and ETFB1, or in the electron transfer flavoproteins ubiquinone oxyreductase genes ETFDH. In patients with this deficiency, all in the mitochondrial flavoprotein dehydrogenase are defective with a specific biochemical phenotype for multiple acyl coenzyme A dehydrogenation deficiency. Yet, in a few patients who have a deficiency that is similar to multiple acyl coenzyme A dehydrogenation deficiency, or no mutations are identified in ETFD, ETFA, ETFB, or ETFDH. Another video.
Lifestyle medicine is the use of lifestyle interventions. Lifestyle medicine is the use of lifestyle interventions directed at the treatment, management, and prevention of disease.
Hey folks, Dr. Keel here, and I want to take a few minutes to talk about riboflavin and
The
its
video
potential health
are
benefits.
of the benefits of
Riboflavin is a water-soluble vitamin, also known as vitamin B2, that is typically included as part of a vitamin B complex. The name comes from the term ribose, which is a type of sugar, essentially, and flavin, which means yellow. Riboflavin is essential for the proper development and function of skin, maintaining the lining of the digestive tract, healthy blood cells, and many other functions. It's needed to break down carbohydrates, protein, ketones,
okay, that's all about the riboflavin of the sugar, of the vitamin, essential vitamin. Not it's hyposoluble, it's hydrosoluble. Prevents all of that diseases. It's a lot important. It's such important. And another potential benefit is that acne, muscle cramps, psoriasis, eggs, nuts, and deficiency, toxicity, not only toxicity for B2, and recommending daily intake for the benefit and conclusions. It's a water soluble, it's often part of the vitamin complex. The vitamin B complex is so so large. B1, B2, B12 is so important. Okay. The riboflavin is the precursor of flavin, a dinucleotide, FAD. The cofactor from electron transferring flavoproteins and electron transfer flavoproteins ubiquinone oxyreductase. We thus suspect an defect in phi-aided biosynthesis or transport and study genes involved in riboflavin metabolism. And another video.
Riboflavin, vitamin B2. It's required for energy, for healthy skin, and the prevention of cataracts. Hi, I'm Dr. James Machino. You know, riboflavin, or vitamin B2, is a yellow-green fluorescent pigment. That's why when you take supplements that have fairly high doses of B vitamins, your urine becomes very bright yellow. That's actually proof that your body absorbed the vitamin, processed it, and then the kidneys filtered some of it, either as an end product of metabolism. It ends up in the urine, and then you see this bright pigment, which makes your urine very bright yellow. But absorption of vitamin B2 increases from 15% to 60% if you have it with meals. And that's true of most nutrients or most supplements. If you take them with meals, the absorption is significantly better. For vitamin B2, from an empty stomach, 15%, have it with a meal, 60% absorption, much better with a meal. The functions of vitamin B2. It's required for energy production in every cell in your body. It's also required for the body to make a very important antioxidant known as glutathione. So glutathione is a premier cellular antioxidant, but it's also an important detoxification agent in the liver. So this is another important role for B2. It's also required for synthesis of steroid hormones in the body, for red blood cell production, and it's required to activate vitamin B6 and the B vitamin folic acid. In other words, if you were to get folic acid from food or supplements, or B6 from food or supplements, but you didn't have enough vitamin B2, the body can't convert those other vitamins into their active form. We've seen this before in people that have certain types of anemia, where they have a low level of folic acid or B6. Giving them folic acid or B6 doesn't reverse the anemia until you give them vitamin B2 as well, because it activates those B vitamins, and then they can play their role in the body in all the metabolic reactions they're required for. Deficiency states of vitamin B2, common ones are things like chelosis. Chelosis means very vertical, a lot of vertical cracks in the lips. Nasolabial seborrhea. Seborrhea, flaky skin in and around this nasolabial fold, an inflamed mouth and tongue. Supplementation studies with vitamin B2 shows that it's very important for prevention of migraine headaches, to help manage cataracts and prevent them, for aphthous ulcers or canker sores. So what I suggest you do is you click on the link below to read the article. Make sure that you're ingesting optimal amounts of vitamin B2. It's a very important vitamin. And to learn how to use it as a supplement, the right doses for various health conditions that you may be.
Okay, let's stop. Mutation. The mutation, two heterozygous mutations, C425GA and C440GA, were identified in solute carrier family 25, member 32, the SLC25A32, and cause the mitochondrial FAD transporter, and inner mitochondrial membrane carries that important FAD from the cytosol into the mitochondria. Another good video.
What do you think of when you think of the word mutation? Do you think about X-Men and some really awesome ability? Or the Ninja Turtles? We loved the Ninja Turtles when we were little. We might be showing our age a little bit. But mutations really are not that glamorous. Most people understand that a mutation is some kind of change. It is a change of genetic material, and more specifically, a nucleic acid. RNA and DNA, they're both types of nucleic acids. So how does a change happen? Remember that in DNA, the base adenine A goes with the base thymine T, and the base cytosine C goes with the base guanine G. And that's all good, but what happens if the wrong base matches up? Many things can cause an error like that. There can be external factors, like chemicals and radiation, internal things too, like something goes wrong during DNA replication, during interphase. Interphase is a stage that prepares cells to divide during mitosis. And now while these things can increase the chance that a mutation is going to happen, it's important to understand that mutations, they're random. If a mutation is going to be a helpful thing for an organism, which is rare, very few mutations are actually helpful, it can't just will itself to get that mutation. An organism can't just will itself to get this. It's definitely not like X-Men either. And more about that when we get to natural selection. Many mutations are actually neutral in their effect, meaning they neither help nor harm an organism. And some mutations are harmful. So we're going to talk about the different kinds of mutations. First, we're going to talk about gene mutations. So DNA makes up genes, and genes code for proteins that influence or make up different traits. So when DNA has changes, otherwise known as a mutation, then different proteins can be produced, and this can affect an organism's traits. So let's look at the gene mutations. So first, substitution. That means you have the wrong base matched. So instead of A with T, you could put A with G. Scandalous. You can have insertion. That means an extra base is added in. You can have deletion. That means a base is removed. Insertions and deletions have the potential to be especially dangerous. Because remember, in protein synthesis, we talked about how bases are read in threes. Well, if you add a base or remove a base, suddenly the number of bases total has changed, right? So if you read in threes, depending on where it happened, everything that's read afterwards has the potential to get really messed up. We call this a frame shift. Now, these were...
Okay, enough. Conclusion. In our patient with ample insufficiency of SLC25A32, a deficiency is fed FAD mitochondrial transport chaos late onset exercise intolerance associated with abnormalities in the acylcarnitide profile. The patient's symptoms appear to be highly responsive to oral supplementation with Riboflavin. Our findings underscore the importance of studying genes involved in riboflavin metabolism in such patients and expand knowledge of the spectrum of inborn error of riboflavin transport. Okay, and the last video and the last part of this video. Oh, wow.
Mitochondria are the fuel refineries of our cells, using food and oxygen to create energy that powers almost every cellular function. Here, mitochondria are localized near contractile units within the muscle fibers, supplying the energy for muscular work. Within the mitochondria, a complex dance of molecules harvests the chemical energy from sugars, fats, and other nutrients. While generating power, mitochondria accidentally produce toxic molecules known as reactive oxygen species. Occasionally, these toxins react with the mitochondria's own DNA, damaging genes critical to the mitochondria's functioning. This causes the mitochondrion to cease to function normally, impairing cell function and becoming toxic to the cell it's meant to fuel. The dysfunction of those cells then contributes to the diseases and disabilities that plague those specific tissues with age. Mitochondrial mutations are especially important in the neurological disorder Parkinson's disease and the age-related loss of muscle strength and quality that even active people suffer. But what if there was a backup to the mitochondrial DNA? Using gene therapy, mitochondrial genes could be delivered to cell nuclei, where genes are kept safely away from the more violent cellular processes. When there's a mutation in the mitochondria, replacement proteins would be supplied by the backup copies of the genes. Fortunately, our cells are already equipped to do this. Only a few critical genes are housed in the mitochondria. 99% of all mitochondrial proteins are actually already delivered this way, from genes that are naturally housed in the nucleus. The cell will simply use this same machinery to deliver the few missing proteins that the backup genes encode. With all the missing proteins now replaced by backup versions, mutant mitochondria would still function normally and would no longer be toxic to their host cells. These cells could then return to health and normal function.
And that's it. Oh, the last part of this video. Goodbye.